Hello, today we are going to discuss mitochondria. Mitochondria is actually the powerhouse of a cell or a battery pack which resides in a cell, providing the much needed energy for the cellular metabolism. Uh, This lecture will be structured around the following subheadings. First happens to be the introduction. Second will be structure of human mitochondria. Third will be functions of human mitochondria. Fourth will be biochemical reactions in mitochondria. Fifth will be role of mitochondria in diseases. Mitochondria is a very essential organelle rather. It is considered to be, as I have already told you, a powerhouse of the cell. It plays a very critical role in energy production and regulation of cellular metabolism. Its origin seems to be rather interesting. It was hypothesized to have evolved from a free-living protobacterium that was engulfed by a primitive host a couple of million years ago. Thus, it's a perfect example of a symbiotic relationship within the cell. So, this theory is really an eye-opener. So, each of the intracellular organelle were phylogenetically distinct living organisms that were engulfed, taken into the cell to become part of it so that both of them can exist in a symbiotic relationship. Now, let us dwell a bit into the key stages of evolution of mitochondria. This is actually a very interesting topic for those who are curious. The various stages of evolution of mitochondria includes endosymbiosis, energy production specialization, gene transfer, maternal inheritance, and continued evolution. Now, what are all the evidence that exist to prove what the theory of evolution of mitochondria? Mitochondria have their own genetic material. I repeat again, mitochondria has their own genetic material in the form of DNA. And the DNA inside the mitochondria is circular in shape, more or less resembling the DNA found inside the bacteria. Whereas DNA inside the human cell nucleus happens to be a linear one. So this DNA is different from that of a human cell DNA, which is seen inside the nucleus. Then reproduction. Mitochondria is known to reproduce. I repeat again, mitochondria is known to reproduce by binary fission, by cutting itself into two, which is more or less akin to that of the bacterial reproduction. And then the presence of a bilayered membrane. The inner membrane, which is considered to be a original bacterial membrane, on the outer membrane is supposed to be the host cell membrane that has engulfed it and taken it inside. And they have their own ribosome. I repeat again, mitochondria have their own ribosome, which is 70S type, which is similar to that of all the bacterial ribosome. So this type of ribosome or mitochondrial ribosome inside the mitochondria is different from the larger ribosomes present in the host cell cytoplasm. And these points goes a long way to prove that mitochondria is similar in size to the free living bacteria. Now, endosymbiosis, the process wherein mitochondria and the human cell happens to coexist in a perfect symbiotic relationship, is known as endosymbiosis. This term indicates a unique relationship where one organism that has been engulfed, taken inside the cell, lives inside the cell and lives in perfect harmony. The common principle governing this aspect is mutualism, where both organisms benefit mutually by their relationship. The eukaryotic cell, which is supposed to contain nucleus and intercellular organelles, with their membrane-bound organelles, evolved per se through the symbiosis, endosymbiosis. This relationship would also be parasitic. But luckily for humans, human mitochondria does not exhibit a parasitic trait because they have evolved much before human beings started their evolution. The concept of parasitic endosymbiosis are also seen in other organisms like Plasmodium falciparum malaria and Toxoplasma gondii, where the epicoplast, which is actually a modified organelle in parasites, which are nothing but photosynthetic alga that has been taken into the cell of Toxoplasma gondii or Plasmodium falciparum. They have evolved as a secondary endosymbiosis because these algae have lost their photosynthetic functions after getting into the parasitic cell, and they have converted themselves into an organelle that synthesize lipids. So this synthesis of lipid is very important for the sustenance of the parasite. So this presence of epicoplast helps in developing newer drugs in the treatment of diseases like malaria and toxoplasmosis. Now, let us dwell a little bit into energy production specialization of the mitochondria. Mitochondria, as I've been telling again and again, generates the energy that is needed for the host cell for the normal functioning and existence. This energy is released through or generated through oxidative phosphorylation. The host cell, on the other hand, takes this energy, consumes this energy, and becomes most complex and also becomes specialized. Next thing happens to be the gene transfer. A significant portion of the mitochondrial genome was transferred to the nucleus of the host cell. I repeat again, a large portion of the mitochondrial gene was transferred from the mitochondria into the nucleus of the host cell. So this actually allows the host cell to exhibit certain amount of control over this organelle, functionally as well as structurally. But even currently, this process of gene transfer still continues, even though at a lower rate. Next interesting aspect which I would like to stress upon is the maternal inheritance aspect of mitochondrial DNA. The mitochondrial DNA is actually inherited solely from the mother because egg cell contains mitochondria while the sperms don't contain mitochondria. So the fetus or child takes or inherits mitochondria from the mother. Egg contains the mitochondria, whereas sperm does not contain mitochondria. So maternal inheritance of mitochondria is very, very important. That's the only way a child can get a mitochondria is from its mother. So all the mitochondrial decided diseases or genetic traits gets always transmitted from the mother to the child, not from the father to the child. Now, structure of the mitochondria. This is actually a double membrane structure which has three important distinct compartments. The outer membrane happens to be really smooth and it is freely permeable to small molecules as well as ions because it contains certain specialized proteins that facilitate the transfer of small molecules and ions These proteins are known as porine. Porine proteins help in the transfer of ions, small ions, into the inner compartment. So the outer membrane is very smooth, freely permeable to small molecules and ions because of the presence of porine proteins. The intermembrane space, the space that exists between the outer and inner membranes, contains a fluid more or less similar to the intracellular fluid or cytosol, and the molecular concentration of that fluid also resembles that of cytosol. The inner membrane of the mitochondria is thrown into numerous folds, and these folds projects internally, and these are known as cristae. This membrane is highly impermeable, and the presence of these folds increases the surface area for chemical reactions that could lead to ATP production. It also contains proteins for electron transport chain, ATP synthase, and specific transporters for metabolites. The matrix, which is a fluid-filled space which is enclosed only by the inner membrane, contains a gel-like fluid. This fluid is concentrated by a mixture of enzymes that are required for Krebs cycle, mixture of enzymes that are responsible for the Krebs cycle, as well as mitochondrial DNA and ribosomes. So the matrix fluid space contains enzymes that play a role in Krebs cycle, as well as mitochondrial DNA and ribosomes also are present in the matrix. This is actually a diagrammatic representation of the ribosome. You see the porine protein, small gateways wherein the ions can get into the can pass through the outer smooth membrane, and the inner membrane is, you can see the folds of the inner membrane or the crista, and inside the inner membrane is the cytosol, which contains ribosomes, circular DNA, ATP synthase enzyme is seen in the cristae, and then electron transport chain could also be clearly seen in the cristae, and the three spaces also can be clearly seen. You take some time to see the diagram, and you can also draw this diagram to ensure that you get a good understanding of the structure of mitochondria. Now, let us dwell a little bit into the functional aspect of mitochondria. Mitochondria is known to produce ATP. It is known to cause programmed cell death or apoptosis. This is responsible for destroying cells because unbridled proliferation of cells can cause tumors. So apoptosis ensures that the number of cells inside the organ is kept in check, so that there is no abnormal increase in size of the organ. Then it also plays a very important role in called calcium homeostasis. Then it is responsible for the heat generated inside the cell. In other words, the body temperature owes its presence to mitochondria, which happens to be the powerhouse. Then it plays a very important role in regulation of the normal metabolism of the body. Now, the first function happens to be ATP production. So we will dwell a little bit into ATP production function of the mitochondria. Mitochondria produces ATP by a process called oxidative phosphorylation. This involves an electronic transport chain mechanism and chemiosmosis. I repeat again, the mitochondria produces ATP by a process called as oxidative phosphorylation. This involves an electron transport chain that transport electrons, and then chemiosmosis. Electrons, in fact, they form molecules. These electrons come from the NADH molecules, that is nicotinamide dinucleotide plus H molecules, and FADH2, flavin adenine dinucleotide. So electrons are generated from NADH and FADH2. They are passed along a series of protein complexes in the inner mitochondrial membrane. That is happens to be the crystal area of the mitochondria. And this pumps proton into the intermembrane space, namely the space between the inner and outer membrane. When this happens, there is a proton gradient. This proton gradient drives the ATP synthase enzyme, which is also present in that area, to produce large amounts of ATP from ADP and phosphate. So this is where energy is unlocked and sent in the form of ATP. The next aspect or function that is related to the energy generating mechanism of mitochondria happens to be the electron transport chain. This process begins with the transfer of electrons from molecules like NADH and FADH2, which are generated during the process of Krebs cycle in the mitochondrial matrix. So inside the mitochondrial matrix, I've already told you, all the enzymes necessary for the Krebs cycle are available, and it converts glucose, burns glucose, releases these electrons. They get transferred from one protein complex to another inside the inner mitochondrial membrane. Next comes the proton pump, because as these electrons move through the chain, the released energy that are used to pump hydrogen ions from the matrix into the intermembrane space. So these electrons, when they move through the chain, it releases energy and stimulates a pump to pump proton into the from the matrix into the intermembrane space. Now this is where oxygen comes into play. Oxygen has an important role here. It acts as a final acceptor of the electron. So this ends the oxygen, presence of oxygen here ends the chain of oxidation reaction. Now next stage is chemiosmosis and ATP synthesis. So this process of chemiosmosis and ATP synthesis can be discussed under four headings, or four headings, whichever you choose to term it. Number one happens to be the proton gradient. This is caused due to the pumping of protons. This creates a high concentration of protons in the intermembranous space, that is between the outer and inner membrane of the mitochondria. It forms a gradient, electrochemical gradient across the inner mitochondrial membrane. The next heading happens to be ATP synthase. This gradient, which is formed between the inner and outer mitochondrial membrane, is a potential energy, and that is used to power or stimulate the ATP synthase enzyme. This ATP synthase enzyme is present right through the inner membrane. Then comes the third aspect, or the third head, that is the proton flow. Protons again flows back into the mitochondrial matrix through ATP. Protons flow back into the mitochondrial matrix through the ATP synthase, and this flow provides the energy for the enzyme to synthesize ATP. The last head happens to be ATP production. The ATP synthase uses the energy from this proton movement to combine ADP with inorganic phosphate to create ATP, which is the ultimate energy molecule. Now, let us discuss a little bit about the biochemical reactions inside the mitochondria. Inside the mitochondria, biochemical reaction takes place. It makes the usable energy, generates usable energy by turning food and oxygen into ATP. So mitochondria biochemically converts food and oxygen into ATP. It follows a stepwise flow of electrons that drives the ATP synthase to pack energy into ATP bonds. The mitochondria can be, in fact, compared with that of power plants, where fuel from food is broken down to make high energy carriers like NADH and FADH2. Food is broken down into high energy carriers like NADH and FADH2 that feeds electronic transport chain which are present in the inner membrane, and then it powers or stimulates ATP production inside the cell. Cells that are highly active like that of cardiac muscle as well as brain has large number of mitochondria to sustain their energy requirements. And the next thing that happens to be the power line of the mitochondria, that is the inner membrane. I have been stressing the most important aspect of the mitochondria is the inner membrane, where these electrons run through series of protein complexes which are present in the cristae of the inner membrane. This stimulates the chain and keeps the entire chemical process going, ultimately leading to the development of ATP. And now let us dwell a little bit about the various diseases that these mitochondria are involved in. So mitochondria by nature, I have already told you, is the powerhouse of these cells. So mitochondrial diseases, in fact, most commonly involve those cells that have huge power requirements, like brain cells and cardiac cells, where if mitochondria does not work properly in a brain cell, it leads to Alzheimer's disease. Alzheimer's disease is a classic example of mitochondrial disorder. In fact, mitochondrial disorders have been postulated even in the pathogenesis of diabetes mellitus, and it has also been postulated to be the cause of various liver disorders. So wherever the high energy required cells are present, they can go ahead and create problems when mitochondria is not functioning properly. Thank you.